Hello and welcome to the second of the key areas for Unit 1 of National 5, that is the transport across cell membranes topic. Now, with all these videos, I will move through things quite quickly. I'm just trying to get all the facts out, all the things you need to know. The idea is that you can stop, you can rewind, you can rewatch. Okay, so that's the thinking behind it. We start off the topic by thinking about the structure of the cell membrane. The cell membrane of a cell, as we know from the first topic, is involved in the control of entry and exit of materials. It is composed of two parts: the phospholipids, which are given the flexibility of the membrane, and the other part are proteins. Now, proteins take different forms. This diagram attempts to kind of try and show you that. The main two that we're going to be kind of thinking about today are the pores that are formed by some proteins, which actually give a channel to allow things to move in and out of cells. So hopefully you can understand why that's relevant to things moving through the membrane. And also carrier proteins, and those come in a wee bit later. But fundamentally, the first thing you need to remember is that the cell membrane is composed of phospholipids and proteins. Moving on from that, we then start to think about the different processes by which molecules move. They will move in two directions, possible directions. That is either from a high concentration to a low concentration. That is down what we call the concentration gradient. Concentration gradient is the difference between two concentrations. So molecules will move from a high concentration to a low concentration down a concentration gradient through two different processes. One of them is called diffusion, where molecules will move in the following way. So if I use a circle to denote a molecule, what I'm going to try and show you is on the left hand side here we have a high concentration of molecules, and on the right hand side here, on the other side of the membrane, a lower concentration. Now what I'm telling you at the moment is that by diffusion, molecules will therefore move from the region of high concentration to low concentration, so molecules will move across the membrane in that direction. If those molecules are water molecules, then we use a special word for that, but there really is no difference between the processes. But the special word is osmosis. So osmosis is the special diffusion of water molecules. But in both cases of diffusion and osmosis, they are both said to be passive processes. They do not require any energy. Naturally, molecules will try to move down the concentration gradient that we have just shown you. On the other hand, though, in some situations, the molecules will actually move in the opposite direction. They will be moving from a low concentration to a high concentration. Now, because of that, because they're trying to move against the concentration gradient, that requires an input of energy. But it also requires, I'll just stand to the other side of the diagram, but it also requires, instead of just the pores, it will actually start to involve the membrane proteins that we talked about earlier, the carrier proteins. So these carrier proteins actually take, usually it's ions actually you see in things like questions and stuff about it, but they're taken against the concentration gradient. So molecules are moved from a low concentration to a high concentration, and that process that I'm describing, as I said, is called active transport, and active transport requires energy. So that's one of the very fundamental questions, is that you are asked what's the differences between diffusion and active transport. So the fundamental differences between diffusion and active transport is that diffusion is a passive process, so does not require energy. Active transport does. And diffusion, molecules move from a high concentration to a low concentration along the concentration or down the concentration gradient, whereas in active transport they move from a low concentration to a high concentration, which is against the concentration gradient. So those are the two differences. I know I'm moving fast, but the whole idea is you listen to this a couple of times, you rewind it, you stop it, you pause it. A couple more things just to get ahead, get across to you. Remember that we talk about membranes being selectively permeable. What that means is that some molecules can pass through the membranes, others can't. Things like oxygen and glucose are small enough, sufficiently small, that they will move into the cell by diffusion, whereas other molecules like starch, like protein, they are too large, they do not fit through the pores, and therefore they can't pass through, which is why we call them selectively permeable. Water will move in various directions, which we'll come to just at the very end of the video. And the other thing to talk about would be the waste product of animal cells. Carbon dioxide would generally diffuse out of the cell. Now, that's quite helpful to think about, because what happens inside a cell when it respires is carbon dioxide starts to build up, so it will maintain that high concentration inside the cell, which will actually encourage it to diffuse out. Similarly, oxygen and glucose, they'll be getting used by the cell all the time, so it will maintain quite a low concentration, and therefore the oxygen and glucose will be encouraged to move into the cell by diffusion. Alright, so the only thing that really remains to talk about, because we've talked about the structure of the membrane, we've talked about diffusion, osmosis and active transport, we've used the term concentration gradient and selectively permeable. Really the next thing to talk about is the process of osmosis in a little bit more detail. So this area of the board is where we're concentrating on now. When an animal cell is placed into pure water, the scenario is that the high water concentration is going to be outside of the cell. So if there's a high water concentration outside the cell and a comparatively low concentration inside, water is going to move in and the cell is going to burst. It bursts because it lacks a cell wall. If you remember, that's what a cell wall offers a plant cell that animal cells don't have, is that protection from bursting. Similarly, if you put it into a very strong salt solution or a very strong sugar solution, comparatively, the high water concentration is now going to be inside the cell, so water is going to move out and the cell will shrink. Now, exactly the same scenario is true with plant cells, except because they do have cell walls, instead of bursting, the plant cell becomes turgid, and instead of shrinking, the plant cell becomes plasmalized. But the rule is exactly the same. If you place it into pure water, water will move from a high water concentration to a low water concentration and move into the cell. But if you place it into a strong salt solution, the high water concentration is going to be inside the cell, so therefore water is going to leave the cell. So animal cells will either burst or shrink. Plant cells will either become turgid or plasmalized. So just make sure you're happy enough with that language. Similarly, some questions will show a beaker which has perhaps 1 gram of potato tissue in it, and you might be expected to say, well, what happens to that 1 gram of potato tissue? So all you need to remember is if it's placed into a solution with a higher water concentration than itself, then water will move in and it will gain mass. And similarly, if you put it into a solution which is so strong that the higher water concentration is actually inside the potato, then it will lose mass, and that's something that you would have to know, describe, and actually explain the reasons for as well. I think that covers everything. Thanks.